So welcome to Mendelian Genetics Part 2, and in this class we'll think further about the Rebop experiment that we spoke about in Part 1. So if you remember from yesterday, we had two parents, Robert and Samantha, which we can shorten to Bob and Sam. And Robert, the father, had two dominant genes, which we write as big G, and these were genes for his snout colour, and his snout colour was green. But Samantha, the mother, she had two recessive genes, small g, and her snout colour was the recessive trait, which is red. We can call the genes that they have there for this trait their genotype. And genotype, according to our textbook, is defined like this: the genetic makeup of an organism. So the genotype of Bob for the stout colour genes is big G. Now, what they look like, their physical appearance, we use the word phenotype. And phenotype is defined as the outward physical appearance of an organism, or it could be some function as well. And remember, the first generation, I gave offspring which all looked similar with respect to their snout colour. So this is one of their offspring, which we'll call Toby, green snout, and the genotype of this individual is big G, small g. And because of this dominant gene, the phenotype is this green snout. Another Rebop trait is tail colour. So here big B is the dominant allele, and that results in a blue tail, and the recessive allele is small b, and that results in a green tail. So if you saw an organism like this, an individual like this, a blue tail, what are the possible genotypes? There are two possibilities. One is that this organism is big B, and another possibility is that he or she is big B, small b. In other words, in this case having two dominant alleles, or in this case having one dominant and one recessive allele. And an organism which has two identical alleles, like this one, big B, is said to be homozygous for this allele. And an organism which has two different alleles, you can probably guess, is said to be heterozygous for this gene. Of course, there's another possibility for the homozygous form, which is to have small b, which we would say would be homozygous recessive. So we thought about a problem, which is if two individuals who are heterozygous for the tail colour gene have 10 babies, how many green-tailed ones they will have. So in other words, their genotypes are big B, small b, and one parent, and the other parent is big B, small b. How many green-tailed offspring will they have? And some people were able to correctly predict it maybe around 3. So on average we would find something like that about 2 to 3 offspring out of 10 would have green tails. But how can we predict that? Well, one way to conveniently predict the outcomes of genotypes and phenotypes in genetic crosses is to use a Punnett square. And in a Punnett square, the letters representing the alleles from one parent are written along the top, and from another parent are written down the side. So with these two parents, we have the alleles from one parent here and the other parent here. And then we can write down the genotype of individuals which will be produced when these alleles combine. And we get something like this. And if we remember that the dominant, if an individual has a dominant allele, it will have a blue tail. If it has two recessive alleles, it will have a green tail. We can see that in this cross, approximately 25% of offspring will have green tails. This is a practice problem from our textbook. So in peas, the yellow colour, determined by big Y, is dominant to the colour green, which is determined by the small y. So according to this Punnett square, what is the probability of an offspring being yellow? Well, we can see that the ones which are yellow have the big Y dominant gene, dominant allele. So the probability is 75%, 3 and 4. And the next question, what is the probability of an organism having the small y genotype? That probability is 1 in 4, 25%. And that organism, that plant, would have the green pea colour. And since we're talking about peas, I have to say that, of course, the father of genetics was actually Gregor Mendel, who was an Austrian monk who experimented with peas in his monastery garden. It wasn't someone experimenting with rebobs. It was actually Gregor Mendel experimenting with peas. And these are some of the traits which he looked at in his pea plants, showing dominant and recessive alleles. But the ideas that he discovered are exactly the same as what we spoke about in our discussion of rebobs. So we can sum up the principles of heredity. Traits are controlled by genes which are found in chromosomes, and alternative forms of the same genes are called alleles, and alleles can be dominant or recessive. And when a pair of chromosomes separate during meiosis, the different alleles go into separate sex cells.